cells, and this so-called DTP cell population may be the source of the cancer relapse. And therefore, it would be very important to explore the basic biology and clinical features in cancer therapy, including targeted therapies. And since cancer stem cells or DTP cells are considered as root of primary and acquired therapy resistance, this should be an important question and discussed in the present webinar. I'm trying to get my next slide, which is not really working here. Yeah. So after this short introduction, I would like to talk about the selection and targeting of cancer stem cells. The first introduction of this cancer stem cell hypothesis was by John Dick at the Ontario Cancer Institute in Toronto in 1994. So it's rather early. And he had several papers with this hypothesis and repeating this idea, which says that cancer stem cells comprise a unique subpopulation of neoplastic cells within tumors that is highly tumorigenic and relatively resistant to standard therapy. So again, a subpopulation with special tumorigenic and resistance capacities. So what do we call cancer stem cells? So these are self-replicating cells within tumors capable of regenerating the given malignancy. So it's focused on one single malignancy, characterized by a relatively low number, but this very low number, even one cell, is capable to regenerate the tumor. Moreover, these cells can produce tumor colonies both in vitro and in vivo, and show chemotherapy and radiation resistance, which involves slow division, drug transporters, mutant enzymes, higher mutation rate, etc. Well, that's a very complex idea, and I will try to show that some of these issues are contradictory. But what's the idea how tumor stem cells are generated? So one possibility that these tumor-generating mutations occur in normal stem or progenitor cells, or in the developing tumor, some cells regain stem cell properties. So the two ideas are shown here. So how cancer stem cells are generated. Are they generated from stem cells? So there are even pluripotent and other kinds of stem cells, and they are just dividing and producing again normal stem cells. But some of them are mutated and go into a tumor-like proliferation, and they generate the cancer stem cells. Another possibility that tissue-derived progenitor cells are dividing, and they are mutated and become cancer cells, and again become the cancer stem cells. But it's even possible that differentiated cells can be de-differentiated, and self-renewal would occur, and again they would produce the cancer stem cells. This latter idea is supported that by now we know that all differentiated cells can be reprogrammed into stem cells. So the next slide, which comes very slowly, but hopefully it will come. It's not working now again. somehow I have to stop here because this is now not really working. Okay, this is our next slide. So then how do we imagine that cancer can be eradicated? So we can have the tumor with the so-called cancer stem cells. And if we unspecifically target all the tumor cells, that the tumor will shrink, but the self-renewing cells will remain alive and a relapse will occur. Now, if we can target the cancer stem cells, they will be killed or disappear, and the tumor will shrink and regress and eventually disappear. Now, it's interesting that at the same time, cancer stem cells are believed to be resistant to chemotherapy. So what happens if you use a chemotherapy, or which just generally kills the cells this way, but the cancer cells, the stem cells survive, then there is a tumor relapse. If you manage to kill the cancer stem cells, there is tumor regression and disappearance. So what could be the role of the multi-drug ABC transporters in this process? I would like to focus on this area of cancer stem cells because chemotherapeutics are the most effective treatment for metastatic tumors. However, the ability of cancer cells to become simultaneously resistant to different drugs is a major barrier to successful chemotherapy. And these multi-drug transporters are able to generate such a drug resistance. So what are these ABC multi-drug resistance? They are present in our normal cells, but they can also be present in tumor cells, and they can potentially be regulated by drugs, hormones, and xenobiotics. So the major role of these ABC transporters that hydrophobic drugs, xenobiotics, or hormones can normally enter the cells through the plasma membrane, but several ABC transporters are able to extrude these drugs from the cells, and the key players are the ABCB1, ABCC1, and the ABCG2 transporters. And whenever these hydrophobic agents enter the cells, they can regulate the expression of these transporters by transcription, translation, processing, trafficking, or destruction. So in addition to their presence, they can be regulated by complex ways. Now, the key multi-drug transporters, ABC multi-drug transporters, are the ABCB1, the ABCC1, and the ABCG2 transporters. The ABCB1 protein can transport all kinds of hydrophobic drugs, and the C1 and the G2 transporters can transport both hydrophobic drugs and conjugated metabolites. So these three major transporters can handle thousands of substrates, and many of them are actually anti-cancer drugs. And these can be transported with an overlapping specificity by the MDR1, MRP1, or the ABCG2 protein. So whenever you use a chemotherapeutic drug, you can expect that it will be transported by one or the other of these multi-drug transporters. And although you can achieve successful treatment of cancer with targeted inhibitors, usually resistance also develops. And one of the key causes of this drug resistance is the presence of the multi-drug transporters. So when these transporters are present in tumors, it's interesting that during their discovery, in drug-selected cancer cell lines, there were complex gene rearrangements, gene amplifications, multiple chromosomal copies, but it has turned out that it's not the real world. It almost only occurs in the laboratory. In the clinical cancer drug resistance, the MDR ABC transporters are also important, but they have only low levels, either ABCB or ABCG2 transporters. Very low levels can cause already a drug resistance, and there is no mutation, no gene amplification in the clinical tumor samples. So what can be the background of this defense mechanism? Selection of MDR-ABC expressing cells or induction or regulation of these transporters in the tumor stem cells. And this is just a slide showing that if you have in a given cancer MDR positive cells, usually the survival is much less than without the presence of these proteins. So how they are related to stem cells? It has been first shown in hematopoietic stem cells that there is a so-called site population. So if these cells are incubated with a host dye, some of the cells actually extrude this dye, and these are called the site population cells. And in the site population, if you separate them, then the major protein which is present is the ABCG2 transporter. So it's highly expressed in the site population and practically not expressed in the non-site population cells. So how does it relate to the tumor cells? Well, there are many examples now that the site population is also present in the tumor cells. And this is again related to the high-level expression of the ABCG2 protein. And if you inhibit this protein, then the site population disappears. So it's a functional protein which is present, at least in some tumors, in the stem cell population related to the site population. So how does this ABCG transporter relate to cancer stem cell resistance? So if it's present in cancer stem cells, although the physiological expression are in tissue barriers, stem cell, normal stem cells, and it's related to the removal of metabolized xenobiotics and protection of stem cells, if it's expressed in cancer stem cells, then it protects the tumor cells, and it's because it shows a promiscuous recognition of various drugs and xenobiotics. It shows an MDR phenotype. So one idea would be to eradicate the cancer stem cells, and that we called in some of our papers a stem kill approach, which means that you want to generate anti-cancer agents, whether they are targeted or general chemotherapeutic agents, which show selective ABCG2 interactions. So if they are transported by ABCG2, they become ineffective agents, especially in the cancer stem cells. If you generate anti-cancer agents which can evade ABCG2, that is, they are not substrates of this transporter, they are able to kill these cancer stem cells. Another possibility, effectively inhibit ABCG2 by some of the anti-cancer agents. And in this case, in addition to an anti-cancer action, that will inhibit the transporter and even promote the action of other ABCG substrate cytotoxic agents. So what could be the clinical relevance of the MDR-ABC transporters? So during cancer development, whether it comes from stem cells or already differentiated cells, some of the cancer cells, we can call them cancer stem cells, have expression, relatively high-level expression of these MDR proteins, especially ABCG2. And during transformation, mutation, induction, they undergo partial differentiation and they lose this transporter expression. So when you are using chemotherapy, then you cannot kill these cells which have MDR expression, and you can kill all the other cells which do not have this. And these cells can be selected either based on mutations, induction of the proteins, or just selection. And when you have the relapse or the metastasis, already these cancer stem cells containing the drug transporters are appearing. So actually, this is an induction of cancer stem cell growth by chemotherapy. Now I will finish my presentation with the idea which are shared by many clinicians. Then the question is how to eradicate cancer and the cancer stem cells. And among gardeners, there are quite a few who think that you are able to eliminate the weeds only if you dig them up by the roots. So if you remove them one by one, then you can eliminate the weeds. But there is another general idea that it may be enough if you want to have a nice garden. It's enough just to mow the lawn frequently, and the grass will survive and the weeds will practically disappear. This idea is related to the transformation of cancer to a chronic disease. So if you can transform cancer growth to a chronic disease and treat them sequentially at the good times, then you can practically eliminate all the bad aspects of the cancer. So I will stop sharing here, or just to go to the next slide, which means that we can have now the discussion of this first talk, if you have any comments from the speakers or from the audience. Please make yourself appear on the screen if you want to share your ideas, or just turn on your microphone. So are there any questions to this presentation? I see one question. Yeah, I just see an encouragement that all questions are welcome here. Maybe I will ask just Gergely Szakács to comment whether you will talk about the potentially resistant cells, not as cancer stem cells, but cells which are surviving the chemotherapy. And
exactly. Thank you, Balázs. Actually, my talk is a perfect continuation of all the themes that you have introduced. And indeed, as you indicated, these cells that are so important for the survival of the cancer cell population, now either termed cancer stem cells or persister cells, are, as I will show in my talk, are also they also express some of these transporters, and I think these transporters play a very relevant role at this stage also. So basically, that's what I'm going to cover in my talk.
Okay, but Yeah, thank you. And I wonder if we have here Hexin Chen, who would be our next speaker. And he could talk about his topic in this webinar, which would be how inflammation promotes cancer stem cell maintenance. I wonder if he's here already in the webinar. I actually don't see him as participant.
Hi, Sakadi. Due some reasons, He Xin is not coming now, and maybe we can move to the next speaker.
Okay, so I will ask actually Gergely Szakás to talk about the drug-tolerant persisters, and that will be related to the previous talk and so on.
All right, hopefully you see my screen now. Is that a yes?
Yes,
Yeah.
we can see.
Okay, so you can see the presentation mode, correct? You see my title slide. Yes? Hello?
Yes, we can see.
Okay, all right. Okay, so basically my talk follows Balázs's introduction perfectly. I also start from this scheme where we start with a sensitive cell, here labeled with the red circle, that becomes somehow resistant over the course of the treatment. There are many different reasons why cancer cells can become resistant, and these are just the cellular factors. Basically, anything that happens to the drug prior to its hitting the target can lead to resistance. And as Balázs has introduced, one major preemptive mechanism is already at the level of the membrane, where these transporters are overexpressed and they kick drugs out of the cells. Thus, these drugs cannot reach the critical concentrations needed for the cell-killing activity. We know that these transporters belong to the ABC transporters superfamily, and as Balázs has introduced, there are three prominent members of this superfamily: P-glycoprotein or ABCB1, ABCG2, and ABCC1 that have been shown to be expressed on cancer cells. So what happens is that perhaps when the therapy is started, the majority of the cancer cells do not express these resistance factors, and when at the time of when resistance is experienced at the patient with the patients, then perhaps these tumor cells are equipped with transporters. But of course, in real life, the situation is much more complex. And if we look at all the possible mechanisms by which a patient can become refractory to treatment, we'll notice that only part of these can be attributed to cellular mechanisms. The cancer cell niche, the microenvironment, is another very important factor. The plasticity of the whole system is a very important factor. And you have all these mechanisms which also play a role. So how can we really study resistance by acknowledging also this complexity? A number of years ago, genetically engineered mouse models have been introduced into the field by Piet Borst's group at the Netherlands Cancer Institute. Basically, these animals develop spontaneous tumors, in that case, triple-negative breast cancer tumors, because of a induced deletion in epithelial cells. It's a BRCA1 and p53 loss selectively in epithelial cells, and as a result, mice develop memory cancer or in about 1 to 200 days. What is really important is that these cancers resemble closely to the human triple-negative hereditary breast cancers. And also from our perspective, it's important that they can be treated by chemotherapy and they respond to treatment. And after some time, as a result of repeated treatments, they become resistant. So initial sensitivity is also shown on this tumor volume curve. On the x-axis, you see days. Each arrow represents a treatment, and the y-axis shows the tumor volume. And as you can see, treatment has some intermediate effect. The tumors can be pushed back, but eventually the tumor becomes resistant. What is important, and this was shown already in the Netherlands Cancer Institute, that the mechanism by which tumors become resistant against, for example, doxorubicin, is again the overexpression of transporters. So these resistant tumors use the same mechanism as was seen so many times in vitro when we, for example, selected cells with doxorubicins in vitro. So this in vivo model, which on one hand is a very good model for triple-negative breast cancer, can be now used to study the evolution of resistance, which is again mediated by PGP. And in this lower figure just shows that PGP activity can be monitored in vivo if you use a tracer, which is a substrate of the transporters. You can see that in tumors that express high PGP levels or ABCB1 levels, the accumulation of this tracer will be reduced as a result of the efflux function. So again, we have a model which recapitulates the evolution of therapy resistance. The mechanism of PG is mediated by PGP, and all these factors, which I in a few slides before I indicated to be relevant in vivo, are also at play here because this is an in vivo treatment protocol. So you can ask the following questions. Can you now design a therapeutic approach to overcome this resistance? Can you understand the evolution of resistance? And if you do, can you prevent it? And in this talk, I will show you that with this model, you can actually ask now new kinds of questions, which not only relate to resistant clones, but also addresses intermediate stages. So recently, the field is getting familiar with the theme of persister cells. And to introduce persistence, I think I have to go back to one of Balázs's slides, where he showed that if you start with a sensitive tumor cell population and you end up with a resistant population here labeled with blue, there are multiple scenarios or models that you can imagine. You can imagine that initially there was some sort of a heterogeneity, and already at the first during the first treatment, you had some cells which were intrinsically resistant. And what you do with the therapy, you just select these out. You can imagine that during treatment, somehow a mechanism is induced as a mutation, and then those cells which become resistant then have a survival advantage, and this will lead to resistant tumors. But there is a third possibility, that you start with a sensitive population, and as a result of a treatment, you have a transitory phenotype, which, however, is lost as the tumor cells start to proliferate again. And this transient resistance or persistence is called drug-tolerant persister cells. And much attention is focused nowadays to understand the mechanism by which tumor cells can endure treatment even without a stable resistance phenotype. And this is, if you think about this, this is really reminiscent of the cancer stem cell idea. So these are cells which themselves are perhaps not tumorigenic, but they provide a source for cancer cells, and thus could be considered the root of the cancer population and that Balázs was referring to. Okay, so again, this slide just summarizes some of the information that has been revealed recently about these drug-tolerant persister cells or DTP cells. First, we know that they survive treatment per definition. Second, importantly, they demonstrate a transient phenotype. So when they start to proliferate, this phenotype is lost. And they can be in a quiescent or dormant state for a very long time, or a slowly cycling state for a very long time. And this is better shown here with another curve that we have in the lab. Again, you have the same mouse model. You have different kinds of treatment. This is a combination treatment, and you can see that after the first treatment, there is a very strong response that endures for more than a month. This is a minimal residual disease stage. You don't measure any tumors, but actually the tumor relapses and is sensitive for a second round of the treatment. So these cells that survive here, again labeled with blue, are the persister cells that we want to study. This recent review that appeared last year summarizes many more characteristics of these cells. So what we wanted to do in the lab is to understand a bit more about these cells. So we established an in vitro assay where we try to mimic this situation. So we take cells, here MCF7 cells in a flask, and we treat them with very high concentrations of drugs, here doxorubicin, for a few days. As a result, most of the cells die, but some survive. If we look at the flask very carefully, we can see single cells that show a very weird, bizarre phenotype that survive. And after sometimes 1 or 2 months, these single cells start to proliferate again, and we have again a new cell line that we call repopulated cells. So we wanted to study these persister cells to understand, their secret. What we this first slide just shows you the experiment again. So this is treatment, persister stage, and relapse or repopulation. What's important is that when we compare the sensitivity of the initial cell line with the repopulated relapse cell line, the sensitivities are the same. We have here 3 drugs, 3 cell lines. So it's not that the relapsing or repopulating cells become resistant. Again, this is a transient phenotype which enables survival in these cells. The repopulating cells are not resistant. This shows you the time it takes for these cells to recover. You can see that if we take, for example, MCF7, we have 3 different kinds of treatments, doxorubicin, olaparib, or cisplatin, and it takes more than a month for these cells to wake up. So it's a very lengthy process during which time the cells are in a dormant state. Okay, so we collected these cells and wanted to characterize their phenotype. And this is a summary slide here. You can see that we could show that they undergo EMT, they show signs of senescence, they show a number of stem cell markers, a number of epigenetic changes. But for this talk, I would like to focus on the expression of ABCB1 or P-glycoprotein, this transporter. We could show that whereas at the start, the cells did not express any of the transporters, here at very early stages, before the drug was removed, day 6, day 7, a small fraction, subpopulation of the cells expressed higher levels of ABCB1 or P-glycoprotein. This shows you the percentage. So when we treated, again, MCF7 cells shown here with doxorubicin, we could show that at this early stage, about 5% of the cells started to express these transporters, whereas in the repopulated cells, again, transporters were not expressed at all. Similarly, when cells were treated with olaparib and other substrate of these transporters, we could see a similar induced expression in a subset of cells. This induction was not detected when cells were treated with cisplatin, which is not recognized by the transporter. So the big question is, you see no expression, induced expression at the persister stage and then no expression at the relapse. So can you do something about it? Is this relevant? Can you exploit the fact that persister cells show a transient expression, an induced expression of PGP? So to address this question, you can deploy an inhibitor of the transporter. And this is a complex experiment that we performed in vitro. We had a condition where we just treated the cells with doxorubicin that I showed you before. A second scenario where we added toriquidar, the inhibitor of the transporters, when the cells were treated with doxorubicin. And two other scenarios where we in the first case, we prolonged the treatment of inhibitors in the persister stage, or we only apply the inhibitor after doxorubicin has been removed. And we wanted to compare the relapse time, in vitro relapse time of these conditions. So this sort of Kaplan-Meier curve shows you the relapse time. And I was explaining you that when we apply doxorubicin, the cells, really a lot of cells die, but after about 40, 60 days, in each flask, you see the relapse of the new cell line, the repopulated cells. So the first flask started to repopulate at day 55, but then until this time, all the 4 flasks that we had in the study were completely repopulated. So when we added toriquidar together with doxorubicin, there was no difference in this repopulating time, indicating that there was no effect of just adding an inhibitor to cells which do not express any transporters. But when we added inhibitor to the cells after the drug has been removed, so at the persister stage, you can see that it took much more time for the cells to recover, indicating that induced expression of PGP in persister cells provides a vulnerability that you can exploit by the inhibition of PGP. So this is what this experiment shows you. I understand I don't have a lot of time. Balázs, please indicate if I'm really running out. I just want to show you that we repeated this experiment in our mouse model also. Again, we have a treatment protocol where we just add doxorubicin to the mice, and if you do that, you can have transient success, but eventually the tumor becomes resistant to treatment. Then we combined doxorubicin with toriquidar again, the inhibitor. This is condition 2. Condition 3, where we sorry, this is condition when we combine. This condition is when we just add the inhibitor alone without doxorubicin, and then we again in this 4th condition we continued the treatment only with the inhibitor without the drug, again to somehow target persister cells. And what you can see here, this is again a complex figure. You can see that when you don't treat mice, this is the black curve, they succumb to, you have to sacrifice them pretty soon because of the tumor growth. The inhibitor alone, the green line, does not make any difference. It's not toxic. Doxorubicin alone, as I showed you, it results in an overall survival of about 60 days because the tumors become resistant. However, if you maintain treatment with just the inhibitor, this condition here, so when you add doxorubicin and then during the persister phase, the inhibitor, you can really prolong the survival of mice, again showing that persister cells that survive treatment without themselves becoming resistant can be targeted by the inhibition of these transporters. So this brings to my summary, which says that therapy resistance is, of course, a major impediment to successful treatment. Of course, resistance is a complex phenotype based on very complicated mechanisms, of which PGP-mediated resistance might be only a subset. And even though only a fraction of cancers express PGP when they are resistant, we could demonstrate that the success of treatment in vitro and also in vivo with the PGP substrate doxorubicin and can be significantly improved if we target DTP cells with PGP inhibitors. The final slide is the acknowledgement slide. So this work was performed in Budapest and in Vienna, and these are the co-workers and some of the funding. And thank you for your attention. I understand that I was really fast. I was trying to tell you a lot in a short time. So if you have any questions that I can answer, I'm here for you. Thank you.
Thank you very much, Gergely. And that was a very interesting talk. And we still don't know what kind of stem cells or DTP cells we have under various conditions, but certainly we learn more and more about these cells, which it should be targeted and removed if we want to cure the cancer. Are there any questions to Gergely Szakács, please? You can indicate it by simply turning on your microphone.
Hello, Dr. Naga. Do you have some comments?
I don't see any hands raised or microphones turned on. So I think we will just go on with the presentations. And my question is if Dr. Haixin Chen is already present and we can hear that presentation of the inflammation and stem cells, cancer stem cells.
Dr. Lapata has some things, so she cannot come in the webinar, and she recorded her presentation, and I will play it for everyone.
Okay, so that's Dr. Laporte, who would be a participant as well, but she is not here either.
Yeah.
So I think we will just finish this webinar by a discussion on the potential role of transporters and oh, so her presentation is here.
Yeah.
But would she present it?
Yeah, so can everyone hear?
So good morning,
So can everyone hear?
Yeah, we can hear you.
Okay.
Okay. So good morning, good afternoon. Thank you very much for having me in this very exciting webinar. I'm come from the University of Milan, the Center for Complexity and Biosystems. We are that was founded with some other colleagues with different disciplines in 2015. I'm working in a complex system, particularly on digital health, and I was training as a general pathologist, but I'm also actually is about more than 18 years that I'm working in digital health using different strategies to do quantitative biology and prediction. So what's the big question that I really, spend a lot of time of my research thinking about spreading of metastases. We know that now the people are not dying for primary tumors, but for metastases. So it would be very important to predict in specific patients, what will happen to that specific patients in his or her future, where the metastases will spread, and other questions like this. Why metastasis is the big problem? First of all, because most of the time the metastatic cells are in inaccessible sites. And second, because of the aggressiveness of metastatic tumor cells. Actually, we worked very extensively on both of these questions, and I will show you some data and results that we got in the recent years. Starting with the first point, spread of the cells in inaccessible sites. We know very well that metastatic process is a multi-step process, and each of these steps should be occur in order to have, to give rise a secondary tumors and to see, a metastasis. What we did, since the cells can spread in inaccessible sites, and the question was, are we able to quantify and to predict in which organs, in which sites the tumor cells, the primary tumor cells will spread. So, and this is of course depending by two different aspects that can play together or not, the famous seed and soil. So is the structures of the blood vessels, lymphatic vessels. In this case, we focused on blood vessels. So the physics that is behind the spreading. And on the other hand, the characteristic of the cells, the aggressiveness of the cells, the adhesion capacity of the cells. So all the other issues that are, of course, involved in this seed and soil. So the environment, or the seed, the structures of the blood, of the circulation. What we did is, as I show you now in this video, we did a 3D simulation. So we reconstruct here. I'm showing the pancreatic cancer, but it's happening. So we did the same for many different primary tumors. So this is just an example. So we did a 3D reconstruction of the circulatory system using MRI images. So 3D images. So I mean, we completely reconstruct the structures, completely, means that we have now a humanoid that recapitulate a human structures. And also, you can see now, you can appreciate this part. And we also recapitulate the blood flow models. So the model simulates hemodynamic linear blood flow equations from veins that you can see in red. Sorry, it's the opposite. Veins are in blue and arteries are in red. So we recapitulated actually the structures and also the hemodynamic laminar blood flow. So completely the physics of the blood flow system. Regarding to the adhesion properties, they were always the same. We didn't change them. So, and now you can see the primary tumors. These are pancreatic primary tumors that are spreading and go around. So this is circulating tumor cells that are released from the pancreatic tumor primary sites, and they are, going whatever they can in these branching points. That mean turn and go randomly in the branching points. And so we simulated actually, the capability, the possible capability and the projection of the tumor cells. And you see here what, they are moving and go around according to the physics actually that is behind. And then what we did is, of course, we obtained some specific sites, metastatic sites, where these cells were more frequently, reached. So they reached the more frequently specific secondary sites. And what we did is to compare our data, the simulating data, with the autopsies. So we got a large data set with a lot of autopsies got from patients, from subjects that died for specific primary tumor. So the primary tumor was known, and then there was an autopsy and was counted in which sites they give rise metastatic metastases actually. And then you can see here a cross correlation, the comparison between our the simulation and the autoptic, the sites that was found in the real life, in real data. And you can see that we got different results according to the tumors. For lung cancer, there is a pretty well correlation, very good correlation between the simulation and the data. That means that the blood flow and the structures of the systems, the blood flow system is very, is very important for the spreading of these tumors for lung, but not for liver, perhaps, and also for other kind of tumors are less, correlated. And that means that in this case, you can appreciate also better in these, in these histograms, you see lung 82%. So, I mean, this is geometry and flow, and in the red, actually in the dark pink, in the light pink, you see seed and soil. So you see that lung, so the geometry and the flow is very important actually, and is matched very well what really happens in the real life. While for liver, no, perhaps, and also for other kind of tumors, but in particular liver and kidney. So that means in that case, it's very important. This specific kind of tumors is very, very important. The adhesion properties, the aggressiveness of the tumor cells, so all the other, characteristic and capability in the mechanism that the cells use to give rise to metastasis. But for lung, geometry and flow is important, is very important, and can be used for predict in, how many cells will give rise metastasis starting from that specific primary tumors. And so now we have a predicted models that for some tumors works well, and we can predict, giving rise the circulating tumor cells. We can predict in which, in which metastatic sites the tumors will give rise a metastasis and will grow, and that is a predicted model. But on the other hand, we have seen that for other kind of tumors, the aggressiveness is due and involves also the environment and the capability of the cells, probably the adhesion properties and other biological mechanisms. So this is the second point. We worked very hard, try to understand and to answer this question, aggressiveness of metastatic tumor cells. And in this case, we investigate a lot the epithelial-mesenchymal transition. This is a key point of the cells. We have seen also in one of the cartoons at the beginning where I shown the multi-step process of metastasis. Actually, the epithelial-mesenchymal state means that the cells can change their state from a polygonal columnar shape, so specific shape with the apico-basolateral polarization. There is a strong cell adhesion, and they are not able to migrate so much, and they express some specific markers like icadherin. But they can, exactly by environment, many different aspects, they can change, and they can go in the mesenchymal state. In this case, you can see that they change completely the shape. They are spindle shaped. They have these focal adhesion contacts, and they acquire the strong migratory potential. And of course, in this case, we have different markers. So this process is, can go in one direction, but also on the other direction from mesenchymal to epithelial states. So it's bidirectional. So it's this process is called by different ways. So like phenotypic switching, so the cells can switch from one state to another according to the environment, according to many different aspects. But you know, it's not so simple like this, because more recently was shown like this transition between epithelia to mesenchymal state is not just from one state to another one so abrupt, but there are also intermediate states. The cells can go through intermediate states, and these intermediate states seems to be very, very important, because the intermediate states seems to give rise metastasis and give, you know, show a much more aggressiveness even with respect to mesenchymal state. So this seems not, you know, this seems, you know, an important lot of questions. And of course, the capability to go in one direction or in the other direction is depending by microenvironment, the tumor cell modifications, many aspects can be. I worked, so my group worked really a lot and still working on switching. We publish a lot and we try to identify, actually we shown and we demonstrated how there is actually a small population inside the bulk of the tumors that we call maybe not properly cancer stem cells, because they resemble some typical biological features of stem cells, but under specific condition by the environment can switch. So from one state to another, so a cancer stem cells can switch to a cancer cells, but also cancer cells can switch to a cancer stem cells under specific condition. And so we worked a lot regarding this phenotypic switching, this capability of the cells to switch the phenotype in order to understand which, you know, in order to define and to understand the possible targets, which targets we can use to stop this process. This is pretty important, because when a cancer cells switch to a cancer stem cells, that's of course can be, you know, related to patients, can be related to more aggressiveness, maybe in the future, if it's not now. And so to stop this process and to understand which are the key factors regulating the phenotypic switching is very, very important. And what we found, so there are many papers, you can have a look now. There is now another one that is just accepted and will be released very soon in 2022 or 2023 regarding this process. But I can tell you that one of the key aspects that we demonstrated, in particular, my group worked a lot on melanoma. So in these kind of tumors, what we found is there is a complex network of macroRNAs that regulates this process, and in particular way, then we identify the macroRNA 222 that seems to be, at least in melanoma, pretty important for this process. Actually, we demonstrated that targeting this macroRNA, these phenotypic switching is inspired, so it's not going to work anymore in a good way. Anyway, you can have a look at the last in these papers if you are interested. But I'm not talking about this today. I don't want to talk too much about the days, but how we used what we did for other stuff regarding the epithelial mesenchymal transition. So since the epithelial mesenchymal transition is a key factor, we also use an in silico approach. And what we did is to recapitulate in silico, again, the complex network regulating the epithelial mesenchymal transition. We started from Stanley models that was released and published in 2015, but was not physiological, because according to these in silico models, it was possible to go only on one direction, from E to M, but not vice versa. And as I showed you before, the process is reversible. So we added the other nodes, and now this network is reversible. So it's possible to go from EM or ME, and the strength of this network is that now I have the capability in vitro to knock out or overexpress one specific gene and see the release, what is the outcome. We published in 2018 on PNS, and what we got with this model, first of all, of course, we explore what happens in this model, starting from a random state, and then update the rule and find fixed points and see what was the outcome. And we found that we found two states, stable state, that was E and M state, as expected, of course, the epithelial and mesenchymal state, but between the two states, we found a lot of intermediate states, not few of them. And that is very important because, of course, as biologists, we are able to define intermediate states using some markers. So we see overexpression, so both, not overexpression, but co-expression of E and M markers in the same cells. But of course, we have a limited markers, and there is, we are, of course, limited when we do just in experiments, because we have limited numbers of possible markers that we can use. But according to our models, so we can switch and on the different genes, and we found that actually there are many intermediate states, many ways that the cells use to go from one state to another one. And these states, and is shown in this part of the pictures, display a high energy. What does it mean? That they are not stable and they are prone to move more, and so to give rise metastasis. This is actually what I've seen in experiments, that, as I said before, the intermediate states are more, metastatic than even the mesenchymal one. Then what we did is to map RNA-seq data on our landscape, and now we are able to map actually the RNA-seq data in our landscape. And you see that, as expected, this is a control, if I can say this. We analyze RNA-seq data from skin and fibroblast skin. All the RNA-seq goes data went on the epithelial state, while fibroblast in the mesenchymal, as expected. So that means that the model is not just a model, but works very well with the data. And we are now able to overlap, the RNA-seq data on the landscape. We also did these experiments. This is another control of our models. We started from cells that were in the epithelial state, and the cells were treated with some inducers to induce the transition from, in this case, from epithelial to mesenchymal state, and there were RNA-seq data, single cells at different time. And so we analyzed and we overlap, as I showed you before, the data on our landscape. And as you can see, we are able to follow the trajectory of the cells that are moving from the epithelial state through these intermediate states, and at the end arise to a stable state that is a mesenchymal one. And we can do exactly the opposite, starting from, in this case, an M state, and they differentiate the cells with in this case with doxorubicin, and follow the trajectory. So this is very important. So we have now a strong instrument that can help us to follow the trajectory of the cells from the E and the M state, and using RNA-seq. We also analyze data, so we project this data on our landscape. This data were single cell transcriptome analysis of primary and metastatic tumors, head and neck cancer. And what they found is there was this, again, intermediate state that was more aggressive, so they were metastatic, and they show these experiments was actually was not an experiment, but was data from patients. What we did again is to use this data and to project on our landscape. And what we found again is this the intermediate states, the patients that show these cells with intermediate states and were also showing higher aggressiveness, where in our landscape were in the high energy state. So according to our our models, they were more prone to give rise metastasis, and was actually what was shown by the paper actually. So then we focus more, so this was actually in one sense the theory and the models. Now we have a fantastic model that can help us, and we can use to predict, and we can use to overlap RNA-seq data and to predict what happens in which states are the cells. And then we use this for breast cancer. But why? First of all, because we know that there are a lot of cases and is the first cause of death for women. So I'm really interested in this cancer for for many reasons. We know that there are 4 different subtypes of breast cancer, and actually this is a percentage. And in particular, there is the subtype, the triple negative breast cancer, that represents about 20% of all breast cancer. But it's very interesting because there is no yet specific drugs. They are now under, trials, some drugs that are released by different pharmaceutical companies. But since there is at the moment no specific drugs, most of the time the patients, as reported by the literature, are treated when maybe after was discovered that was not necessary, and the treatment was even worse than because of the side effect and other impact. So this is an important percentage, about 30%. So it's important when you have, in this case, a scientific point of view, a triple negative breast cancer, to predict in this subject what will be her future if that tumor is more aggressive or less aggressive, because it has an important impact on the therapeutic strategies that the oncologists can suggest to the patients. So we developed a platform using the EMT transition and also using other strategies that is the base. There is also some artificial intelligence for breast cancer prediction, and is the first platform that also is able to release to give a personalized strategy. You can see here the website where you can have more information technically, but I can just show you some data that we recently published using this platform to validate the platform for triple negative breast cancer. Here we published this on Cell System in 2021. So here we analyzed patients with a diagnosis of triple negative breast cancer, and again we analyzed the RNA-seq using our platform, a little bit modified for breast cancer. And you see this is a single patient RNA-seq, and you see that the patients are again are able, to stratified in different part of the landscape, and the more aggressive, these red are mesenchymal one, the blue are epithelial one, and these actually were the patients with worse prognosis and more aggressiveness. Here you see, you can appreciate better these single patients. You see here that we have here the relapse-free survival over time, and about 120 months. And you see that the patients that had a low risk, according to our platform that we called Ariadne, have a higher, free relapse, so there were higher, chance to survive with respect to this cohort of patients with high risk score of Ariadne. So we have now an instrument, these platforms allow to calculate the risk, the probability that specific patients give rise metastasis over time and can be used as a predictor. Then what we did is to compare our platform Ariadne with the tumor immune macroenvironment meta-signature, again for triple negative breast cancer. So is known that there is some possibility to stratify patients according to the immune system, to reaction of the immune systems. You see here there is some possibility to rank, to subdivide patients according to this immune signature, but there are also a lot of intermediate patients that we don't know actually, and the signature is not able, to catch the specificity of that specific, of that specific patients. So it's not working in that case. So then what we did is to use in these patients, in this data set, our platform Ariadne, and you can see that these intermediate patients that are not completely, are not cached by this immunosignature, in this case, Ariadne is able to identify a subset of patients with a high risk and high score of the platform. So again, can be useful even in addition with this immunosignature, in particular in this gray, in the gray part where the signature is not going to work very well. So this is the clinical practice for triple negative breast cancer of Ariadne. So it's possible now to use Ariadne for, understand which patients can be used maybe for chemotherapy or for other treatments. And of course, what we want now to know is to see how works Ariadne with immunotherapy and targeted therapies. And we are very happy to, to open also new collaboration. If you are interested, please contact me. We are very happy, to use our platform for your data and see what happens. These are the people involved in this project. Myself, Stefano Zapperi is a is a physicist, theoretical physicist, and Francesc. He was our PhD postdoc for 5 or even 6 years. I don't remember, so a long time. And now he has an independent position. And we also, me and Stefano Zapperi, believe a lot in this multidisciplinary, interdisciplinary, new field in one sense, discipline, and we have written this Physics of Cancer that was published on Cambridge University Press, and I really, recommend you if you want to, I don't know if you come from biological discipline and you want to understand much more about the new quantitative strategies or vice versa. Thank you so much for your attention, and I hope that was interesting for you.
Thank you very much, Dr. Laporte. It was really very interesting how these concepts can be turned into clinical practice and predicting therapy response and changing therapies. So again, we have a few minutes for discussion. Is there any question from the audience or from the colleagues participating? Gergely? Yeah.
Yes, thank you very much for this interesting talk. Are you still here? She might have left.
It was an interesting way of presentation through another website, but we will see if we can get responses.
Dr. Lapata is not here today for some reasons, but if you have any questions, and you can tap the word to the chatting box, and I will forward your comments or questions for Dr. Lapata, and later if she had some feedback, and I will forward it to you. Yeah.
You cannot
Okay.
have direct questions to her, but if you have comments, then you can do it now.
You know, I was just going to say that I think it would be interesting to test her prediction tools with the mice. We have now perhaps several hundreds of treatment histories with quite detailed omic data. So it would be interesting to see if indeed this EMT signature that they detect is predictive or informative in the mouse models.
Yeah, you can ask Ariadne to answer some of your questions. Okay, I think we should go on now, and I will ask Dr. Haixin Chen to present his talk, and Lori could probably have to share the screen and give her the floor.
Okay, Haixin Chen, Dr.
Yes. Yes.
Okay. So could you please share your screen?
Okay, well, so welcome to the webinar, and we are eagerly waiting for your talk.
Can you see my screen now?
Yes, we can.
Okay, great. So I need to change to the display model. Let's get it. Okay, good.
Yeah,
So
it's fine. Okay.
should I get started?
Yes, please go on. Yeah.
Okay, sorry, I'm being a little bit late. I thought it's the US times. I'm Haixin Chen. I'm going to give a talk. This title, How to Induce Information Promotes Cancer Stem Cell Maintenance and Chemo Resistance. Sorry. Oh, I cannot move my, okay. So the information has been well known to linked cancer. So this link has been established in 19th century by Rutherford Virchow. So inflammation can impact every step of cancer development, from cancer initiation all the way to cancer metastasis. And in the development tumor, we know the immune cells or the immune can play both the pro and anti-tumor tumorigenic roles. So immune cells can secrete many factors to affect tumor growth, tumor mutations, and so on. So that's a few interesting question. How the node cancer is unwound, unhealed wound. So the big question: how the immune invasion is triggered from first place. So there are many reasons to trigger information. For example, information may be from diet, the environment factors, exposure. There may be the chemotherapy that can trigger information. There may be the chronic inflammation, autoimmune disease, and also known the tumor cell itself can also trigger the inflammation as well. So how this information shape the tumor development is not well understood. So for a while, we know there are models to explain how the tumor is initiated. So there are two models to explain the tumor initiation. I'm sure that first a few speakers have introduced this concept. Now, one is called the statistical model. They believe the cancer is initiated from mutant cell, which accumulate serious mutation, lead the cell to uncontrolled proliferation, eventually lead to tumor formation. During this process, the tumor cell continue to accumulate mutation, and then the cells interact with the environment that applies some pressure, continue to select its most aggressive tumor cells. And in this model, the immune cells play a very important role to select the most proliferative and fitter cells to proliferate. But also we are facing this new model. This new model, brought about in 1990s, the cancer stem cell model. Right now, they consider cancer is a kind of bad organ. It's only a small proportion of cell consider cancer stem cell. They can self-renew and deficient to different type of immune cells. Now, for the statistical model, for a while, we think the immune cells just, how to immune cells can play both the anti and pro tumor genesis role. Now, you put an immune cell in this concept, there are two different models, and the immune cell play a different role here. Now, we know the immune cells during the tumor development, the immune inflammation goes through different stages, from the inhibition stage, equilibrium stage, and equilibrium stage. So the first mutation will occur during the tumor development, and the immune cells may be able to recognize this mutant cell, and then both the innate immune cell and deficient immune cell can be triggered to eliminate these cancer cells, and then these cancer cells never become apparent observed. So this is called the inhibition stage. But when more mutation or epigenetic change occur in these cells, some cells maybe become more resistant, and then that's the immune cell can inhibit some cell, but some cell maybe kind of survive a little better. So the immune system and the tumor growth reach the kind of equilibrium, and this equilibrium can be a long time. And eventually, the tumor cell will escape from this immune surveillance and then go up. Now, we'll talk about this statistical model and the cancer stem cell model, how the immune cell, how to explain the role of the immune cells in these two models. Now, for the statistical model, for a while, we think the immune system to promote tumor growth should introduce some mutations because the immune cell can secrete many cytokines to introduce mutations, and also immune cells can secrete many growth factors. We can promote tumor cell growth. That's why the immune cells, even though they can kill some cancer, at the same time can promote tumor growth. But if you put this immune system in a cancer stem cell model, how we're going to explain the immune system in a cancer stem cell growth or generation maintenance? So that's in order to understand the situation. That's how we try to understand some of these questions here. Now, thanks for the previous speaker, Dr. Paula, introduced the breast cancer background. So we are using especially focus on the breast cancer stem cell, especially HER2-positive stem cell. So HER2 is an oncogene belong to the human epidermal growth factor receptor family. It's overexpressed in 20 or 25% of breast cancer. So the breast cancer generally can be divided to, based on the cell marker, can be divided to three major groups. So the ER positive is a hormone positive breast cancer, sometimes called a Numina A or Numina B cells. And then there's a HER2 positive breast cancer overexpressed or HER2 overexpression breast cancer cells. Another one is the triple negative, and there be ER negative, HER2 positive, HER2 negative, and PR negative, triple negative, sometimes called a basal type of breast cancer. So the HER2 type, even though only account for 20 to 25% of breast cancer, but this type of cancer is very aggressive. Even look at the survival curve here. So this is Numina A, Numina B. Basically, this is a hormone positive breast cancer. They have relatively better survival. But another two type of cancer here, the HER2 positive here, and also the basal, sometimes called triple negative, they're overlapping. So they have much worse survival rate compared to another subtype of breast cancer. So there are two different type of study all show similar pattern. So HER2 positive breast cancer is a very aggressive type of breast cancer. So we try to use this HER2 positive breast cancer model to study the relation between the inflammation and the cancer stem cell maintenance or generation. So we chose this model uses MNT-free HER2 translating mouse model. So this mouse model, the overexpressed HER2/neu in the mouse memory epithelial cells. 100% mice develop tumor eventually, but these mice have a long latency, about 200 days latency. So we are interested in how this HER2 drive the tumor development. Now, HER2, if you overexpress HER2 in vitro in a petri dish, you put it in the petri dish and grow this overexpressed gene in normal cells. They never grow tumor, never form tumor cells. But if you grow these mice, they are carrying the overexpression of HER2 in the mammary gland, these mice will develop tumor, but that's a long latency. So the conclusion here, that HER2 overexpression by itself is not sufficient to transform normal cells into cancer cells. Something still need to help this transformation. So this put in the in vivo, that's what thought is environment contribute to this tumor development. So then question is what's the environment factor why these cells become tumor cells in vivo? So we choose a time point here before the tumor development. We get this memory tissues to analyze the gene expression pattern. One of the interesting we found is that a lot of inflammatory gene expressed during this stage, at this pre-malignant stage. So inflammation at this stage is not as surprising because we know the cancer is also associated with the inflammation. But the question is how the inflammation is triggered. So what's the driving force behind this inflammation? Now, the one just thought just the tumor grow, the tumor going to die, and then the cells, all this content released from tumor cell will trigger the inflammation. Now, we'll isolate this, when we see this cytokine expression, most likely we thought it's just, cytokines secreted by the immune cells. That's why we get a lot of cytokine expression in the tumor environment. But at this stage, we don't see obvious tumor yet, so it's just 4 or 6 months mice. Now, so we thought, well, was it possible the cytokine can be secreted by the normal epithelial cells, not the immune cells? So we get a normal mammary gland, we get a transgenic mice and then white mice. We purify the normal epithelial cells and use this bead, and then we did this the gene expression file again. And interesting, we find this the mammary epithelial cells actually express this cytokine compared to this wild type epithelial cells. So this cytokine is not mainly contributed, from this stromal cell or the immune cells. Now, the information we know they can, affect the normal stem cell differentiation or self-renewal. So in normal situation, the stem cell put in the, the leash, the stem cell kind of proliferates, kind of inhibited or they keep in a quiescent stage. So I'm wondering whether this information in this stage, how they affect stem cell activity in a normal mammary gland. So we analyze this mammary gland this stage, we analyze the cellular composition. So the mammary gland is a well-studied study. They can have one single mammary stem cell, they can repopulate the whole mammary gland. So in the mammary gland composition this stage, the normal mammary gland, they are very small proportion of stem cell, only 0.8% cell stem cell. There are small portion of progenitor cell, they're called epigenial epithelial progenitor cell, and only 3.1%. But in this a pre-malignant stage, about a 6-month stage, and then we found these HER2 transgenic mice have a large proportion of progenitor cells, but not expansion of the stem cells. And that gives, we're wondering why this progenitor cell population expanded, not the stem cells. Okay, if the information going to promote proliferation of stem cell, we expect the stem cell population going to be expanded. But actually, we notice a progenitor population. So later, when we analyze the tumor from these mice, actually, the tumor shows exactly same phenotype as these progenitor cells, so not a stem cell population. So that we ask one question: why is the progenitor population, expanded, not a stem cell? Now they are come to the stem cell, cancer stem cell model. So the cancer stem cell model, they just possess their small proportion of cell have a stem cell like ability, but they don't tell you where is the origin of the stem cell. So this cancer stem cell can be origin from normal stem cell, can be origin from this progenitor cell, can also possible from deficient cells. Now we are seeing here, it's just a progenitor cell population expanded. So the question we're going to ask, whether this HER2 is overexpressing the stem cell, and then promote stem cell to proliferate, and then somehow these cells you can screw up, the division screw up, and then we get expansion of a stem cell. Or this is a HER2 overexpression expressed in the differentiated cells, and then they can trigger the differentiation. This cell going to regain this stem cell-like property. That's a question we don't know. But the interest here, we know here this is the progenitor cell population is expanded. To address this question, how does HER2 affect this is the property of these cells? So we use the more in vitro model to better, understand the potential mechanism. So in this case, we use the human normal epithelial cells. So we introduce these HER2 in these human epithelial cells is a 10A cell, epithelial 10A cells. So we see whether these 10A cells, in these 10A cells, the HER2 can drive this differentiation will affect some property of these stem cells. So when overexpressing these HER2 in these epithelial cells, we did the gene expression profile again. Interestingly, we see all this cytokine which is overexpressed in the transgenic mice also overexpressed in this HER2 overexpressing the normal epithelial cells and some 10A cells. So all the cytokine CXCL1, CXL20, L1, CXCL8, L6, interferon and beta. And GCNS will show the inflammatory response gene upregulated, cytokine secretion pathway activated, interleukin-1 production, LCA signal pathway all are activated. So it's kind of a mimic what happened in vivo. So the next question, so how this cytokine affect the tumor cell growth or affect the stimulus? So the first thing we did was say how the cytokine affect the tumor growth, but actually none of the cytokine dramatically affect the tumor cell growth in vitro assay. So next we analyze whether the cytokine affect the stimulus or whether it can introduce the deficiency and then allow this cell to again this stimulus. So we did tumor sphere formation assay, and then based on what the genes profile, we patch all the cytokine chemokines to culture this cell in vitro. We found these 3 cytokines, L1 alpha, L1 beta, L6, and dramatically increase the tumor formation ability. So based on here, control L1 alpha and beta 6. But even though these 3 cytokines can dramatically increase this tumor sphere formation ability, the L1 beta, even at the MR level, L1 beta dramatically upregulated, but at the protein level, we cannot detect this beta expression in the supernatant. So later on, only focus on this IL-1 and IL-6. So since these IL-1 acids can increase the stimulus, we want to know whether this happen in a normal, breast cancer cells. Whether this IL-1, IL-6 also required to maintain the cancer stem cells. So we use a CRISPR-Cas9 system to knock out IL-1, IL-6 in one of the HER2 positive breast cancer cell line HCC 1954. So and then we'll select knockout cell line, and we use HER2 knockdown cell as a control, and then we end as a cancer stem cell population. When this IL-6, IL-1 is knocked down, the stem cell content dramatically decreased, dropped. So from this 80% drop to maybe 0.5, 0.6%. So we did a lot of molecular work to characterize in a single pathway, and it turns out HER2 triggers sequential activation of IL-1, IL-6 in these HER2 positive breast cancer cells. And then this sequential activation seems very important for the maintenance of cancer stem cells. Now, that's why I explain why, knockout one of these gene also dramatically reduce the cancer stem cell contents when injecting this cell into the animal to further confirm this is really required to maintain the cancer stem cells. The parent cell can grow tumor very well, and the knockdown will knock out the cell line, can form a tumor, but efficiency is much lower, and also tumor grow much slower. So, and then the next question was, needs to happen in the primary breast cancer tissues. So we examine the IL-1 expression in the primary breast cancer tissue by tissue array. We find this is IL-1 indeed can be detected in the most many, about 60 to 60% of breast cancer, and the HER2 IL-1 expression correlated with the HER2 expression status, and also HER2 IL-1 positive breast cancer most likely show this the cancer stem cell phenotype based on the CD44, CD24 phenotype. So if you're staining this breast cancer tissue with these two markers, some from breast cancer show this double positive phenotype, some show this most likely breast cancer phenotype, the CD44 positive, CD24 negative, some is more differentiated phenotype. So we see there's a correlation between the IL-1 expression and also cancer stem cell phenotype in the breast cancer tissue. So the next question asks if this is true, the IL-1 is very important for maintaining cancer stem cell, whether we can possible to target this pathway for cancer treatment. We use this is an inhibitor called IRAK inhibitor. IRAK is one of the downstream target for IL-1 single pathways. So we use a small chemical to block this IL-1 single pathway, and then also use a paclitaxel with a chemotherapy, and then we combine these two drugs together to treat the cancer as well. So this is the pre-treatment, the tumor formation, very similar in all these group of mice. After treatment, so this control treatment, the tumor continue to grow, and then use the IL-1 single pathway inhibitor has a no dramatic effect on the tumor growth, seems slightly inhibit tumor growth, and chemotherapy drug definitely can inhibit tumor growth. Combination, we can see they have IL-1 inhibitor and also paclitaxel show some synergistic effect to inhibit tumor growth. And so, inhibit cell growth is not surprising because you combine any two chemotherapy drug can inhibit tumor cell growth. And the problem it is for the typical chemotherapy drug, we know that chemotherapy drug can efficiently eliminate is deficient cancer cells, but it's not efficient to remove the stem cells. So to say whether this the IL-1 inhibitor can effectively inhibit proliferation of cancer stem cell, so we analyze the stem cell content after few cycle treatment. So we have a combination treatment, we have this single treatment. So we analyze this cancer stem cell content after 21 days post-treatment. At this stage, you can see this cancer stem cell population of this here. So this is the control cells, about 1% cancer stem cells, and use this. This is, I think it's not 5.1. So yes, 5.1. So for the IL-1 inhibitor, they can reduce the cancer stem cell content a little bit from 5.1% to 5%. But interestingly, for the chemotherapy drug, we see this population expanded. So it's currently 11.8%. So the chemotherapy drug can eliminate this non-stem cell, but lead to enrichment of stem cell. But if you have a double treatment, this stem cell population can be reduced. Okay, this also shown this is in the quantitative data showing here. So and also we'll follow up after this treatment. So since this cancer stem treatment need to enrich the stem cell, we assume this after treatment, this is a chemotherapy triggered tumor will regrow very quickly. Indeed, as we shown here, after treatment, we'll further follow up. This is a treated mice. For the chemotherapy treated mice, the tumor will recur very quickly and will regain the growth very quickly. But for the combination treated mice, you can see the tumor will maintain the same size for a relatively long time. After treatment, follow for another 20 days, the tumor stay at the same size; they don't grow. So this is the data showing the cancer stem cell target therapy not only can inhibit tumor growth, can also properly delay the tumor recurrence. So I'll go summarize the position I have so far. So overexpression of HER2 memory epithelial cell induce cell autonomous inflammation signaling promotes expansion of the new immunoprogenitor population at the pre-malignant stage. And HER2 induce sequential activation IL-1, IL-6 signaling pathway in breast cancer is critical for maintaining the cancer stem cell pharmacological blockade IL-1 signaling preferentially reduce the cancer stem population, improve the chemotherapy efficacy, and delay the tumor recurrence. So far we can see this HER2, if you put this together, how this HER2 induce tumor development, we see the HER2 expression, there's a first mutation maybe induced promote proliferation or induce a differentiation to generation. This is the progenitor population, and then in these cells, and HER2 also induce this cytokine expression, help to maintain to generate this stem cell population to maintain the stem cell population. But one thing we are missing here, we're still not putting this in the in vivo, because these cytokines definitely going to affect recruited or activated immune cells. How the immune cells get involved in this process has not been understood right now. So that's part of we're still working on right now. So I will thank the member who did this job. Most work was done by Dr. Xiu Liu, who is a postdoc in our lab, and also thank all the collaborators and then the foundation support. I'm going to stop here. Hopefully you have some question.
Thank you very much, Dr. Chen. It was a very interesting talk. And I just would like to mention that with Gergely Szakás, we are old friends of Igor Roninzon, who is also working now at the University of South Carolina. So
yes. Yeah.
Give him our best regards. And of course, now your talk is open for questions or discussion. And if we can stop sharing the screen, then we will see each other. And anyone, of course, can ask questions. Lori, could you stop sharing, please? Or okay, very good. Thank you. So are there any questions to Dr. Chen?
No questions.
I think it's a very interesting concept that the immune system can not only kill cancer cells, but also stimulate cancer cells and actually change them into more proliferating cell types. And that could be a very important point in future therapies as well.
Okay, thank you so much for your comments. And later I will introduce our journal.
Okay, very good.
Sorry. Okay, so can you see my screen? Okay, now I will introduce the cancer drug resistance, and the editor-in-chief is Godfried Peters, and this is our editorial board team, editorial department team. And if you have any questions, please feel free to contact any of us, and we will solve your questions. And cancer drug resistance is an international peer-reviewed open access online journal. Our journal publishes research articles, reviews, case reports, commentary, and letters on pharmacology aspect of drug resistance and its reversal, including drug design, drug delivery, drug distribution, and ciliary drug resistance. And the total publication is 321. Now, this is some data you can see here. Our journal now cooperate with many international cooperation, and this is a media partners of CDR. And regarding the indexing, we have indexed by this databases. You can see in the right blank here. Especially this year in August, we indexed by CI. To get this top achievement without the editorial board member support, now I will introduce our editorial board members. They are 67 editorial board members from 17 countries, and most of them from all over the world. For example, USA, the Netherlands, Italy, United Kingdom, Germany, and other European countries. And for junior editorial board distribution is the same. Our editorial board member from all over the world, most of them from the developed countries. With the support of editorial board member support, CDR published many high quality articles. From here, we can see highly cited articles published from 2020 to 2022. And this year, to achieve a better development, this year we do a lot of changes. Like in the homepage. In the homepage, we can see many important events recently, and it will be upload in time. And at the bottom of the homepage, there are the data of our of other journals' recent progress, like authors, reviewers, published science and articles, reviews, and this data is uploaded in time. From here, we can see it's easier for us to download the PDF version, and also it's very convenient for authors and audience to see the data of downloads and viewers. For this page, CDR encourage all the authors to provide the geographical abstract and to attract more authors and experts. And for webinars from September, we start to hold online webinars to enhance interaction and exchange among scholars. Now we have held 4 webinars now. And for interviews, we conducted 7 expert interviews. We will continue to conduct interview with expert to strengthen the cooperation between expert and editorial team. So if you have the recent advanced research, and welcome to contact us, and we will do the special interview for you and your team, or your recent advanced outcomes. And to get more promotion, we have a lot of international platform like YouTube, LinkedIn, Twitter, and also today's presentation, we will post the video online, also on the YouTube website. So if you have your the privacy data, please feel free to let me know, and I will now publish your presentation online. So also, if you have the content account, please follow us. And the last thing, the 2020 Best Paper Awards has already been set and will announced on January 2023, and we will launch the 2021 and 2022 Best Paper Awards successively. And lastly, thank you all for your great support. And if you have the suitable manuscript related to drug resistance, please feel free to submit for our journal, and the CDR will get the first impact factor next year. Thank you all. Thank you. This is all my presentation.
Thank you very much, Lori. That was really interesting about the journal. And I would like to thank all the speakers and the audience, the participation. And I hope for a lot of people it was interesting, and we could go on with our research and publications with the journal and with the wonderful ladies who were shown on the picture, who are handling our papers and giving us advice about webinars and so on. So thank you very much again, and goodbye. Thank you.
Thank you, Sakadi, and it's our honor to get your continued support. So if your colleagues or your students have the suitable manuscript, please feel free to contact us, and welcome to submit your manuscript. And we will give you priority precisely.
Thank you very much. Goodbye. See you.
Bye-bye.
Thank you for organizing this event. Bye.
Dr. Chen, Hexin Chen.
Yes, I'm here.
Okay. So today you missed the 2 presentations, and later maybe on Monday we will upload the other experts presentation, and at that time I will send you and you can see watch other presentations online.
Okay. So yeah, I thought it's a year's time. First I read the email, so embarrassed. I can't. Yeah.
I also called the phone to your department office, and there's nobody to answer me.
Yeah. It's really a rush. I just first. Yeah. You kind of. Yeah. It's very bad. Yeah. Yes.
It's
Totally
okay.
my fault. Yeah.
It's okay. Thank you so much for your exciting presentation. Thank you so much for your great support.
Okay. Bye.
Bye bye.